Okay, so in 2009 we heard the incredibly alarming news that malaria parasites are becoming resistant to artemisinin drugs. And resistance was first reported in a very small region of Western Cambodia. Now, as Mike said, this is incredibly disastrous because there's no new drugs to replace artemisinin drugs if we use it. And unfortunately, this emerging drug resistance was nothing new. If we cast our mind back to chloroquine, basically chloroquine was introduced in the 1930s, and now chloroquine resistance is being confirmed in around 40 different countries. So chloroquine resistance was first reported in Western Cambodia in 1957, and that was eventually confirmed in the 1960s. We then saw it spread around Asia. It was reported in Myanmar in 1967, and then we saw it spread to the Asia Pacific and into Africa. And basically, a genetic mutation was identified for the basis of resistance, and that was identified in 2000 after about 15 years of work. So what are the consequences of malaria drug resistance? Well, of course, patients are going to take longer to recover from their disease. We're going to see treatment failures so that patients don't even recover at all. We're also going to see a lot more severe malaria, so people who are getting treated for mild malaria may actually want to progress more severe disease swelling. Also, it's very, very important to note that artemisinin resistance essentially means that people are infectious for much longer periods of time, such that that increases the probability of that person passing on the resistant parasite. And of course, there's going to be increased costs to both health services and patients, as you'll see people staying in hospital for longer, and also more people visiting hospitals due to drug resistance. And unfortunately, you'll see an increase in mortality. And what was interesting with chloroquine is we saw kind of in some areas up to a 5 times increase in the mortality attributed to malaria. And one study in Kenya actually found that chloroquine treatment attributed to almost 70% of malaria deaths in a study in Kenya. Now, unfortunately, chloroquine wasn't the only drug. What we saw was drug after drug was repeatedly failed. So here's data from the Thai Myanmar border for the Shockwell Malaria Research Unit. And what this shows is the cure rate on the y-axis. So if the drug works perfectly, that's 100% cure rate. So if you see by the early 1970s, at this point, chloroquine is actually already pretty much useless. But then it continued to decline until we saw pretty much full-blown resistance. But then saw the same with sulfadoxine pyrimethamine, quinine, and then also different dosing regimens of methylquine. So by the time we got into the early 2000s, there was really a big need to find a new drug that would replace these failing drugs. So the drug that was found was artemisinin combination therapy. Now, artemisinin was discovered in 1971 by the Chinese, and it's basically a herb that's been isolated from the Artemisia and the sweet wormwood plant. Now, this drug is incredibly potent. It kills parasites very rapidly. However, it's also got a very short half-life in the blood. So what we do is we administer artemisinin with a more slower-acting drug. It essentially mops up all the parasites that artemisinin drugs have not killed. And then in 2006, artemisinin combination therapy became the first-line drug by WHO in all endemic countries. So as you can see here, there's a physical lag between 1971 and 2006. There's lots of different reasons for that. Mainly kind of suspicions around what the Chinese were doing. And then also basically getting things through clinical trials. So obviously it takes a long time as well. So what we saw then was that the number of ACT treatment courses procured increased from around 11 million in 2005 to a staggering almost 300 million in 2011. And it's that figure there which makes the emergence of resistance incredibly scary. So as I mentioned at the beginning, in 2009, artemisinin resistance was reported. And this is the paper here. So this paper was published by Donald et al. In the England Journal of Medicine. And what they did here is they treated people with artemisinins and measured how long those people took to clear their parasites. So up here on this graph, we have the y-axis, the base of the parasitemia of those patients. And along the x-axis, the hours after admission. And what they found in this study was that patients from Cambodia, indicated in the red and the purple, took much longer to clear their parasites than patients from Thailand, indicated there in the blue and the yellow. So that was our first indication that artemisinin resistance was emerging in Western Cambodia. and suspected resistance is now being reported at the Thai-Myanmar border, more places in Western Cambodia, in Vietnam, and there's also been another report up at the Myanmar-China border. So like I said, it's incredibly alarming that this resistance appears to be spreading. So one question you might be asking is, why does Western Cambodia repeatedly give rise to resistant parasites? Now, essentially, parasite mutations that confer drug resistance emerge after being selected by drug pressure, so suboptimal levels of drug. So you might think, well, artemisinins have been around that long, how could they exert such pressure? Well, in actual fact, artemisinin has been around in Western Cambodia for around 40 years, because China supplied artemisinin to the Khmer region in the 1970s. So in actual fact, there's been a very long history of drug pressure in this area. Importantly, there's also the unregulated use of antimalarial drugs, particularly the use of artemisinin in monotherapy, rather than in combination therapy with those slower-acting drugs. And of course, artemisinin being administered in ineffective doses, such as people aren't taking their medication properly, and also counterfeit drugs are a massive issue in this region. And a study last year showed that around about 35% of antimalarial drugs didn't either contain an active ingredient or enough of the active ingredient to be effective. So essentially what happened was we had these parasite populations which are regularly being exposed to artemisinin without being wiped out. Essentially, the strongest and most resistant ones survived and then went on to basically spread around the population. And there have also been genetic explanations for why resistant parasites emerge in Western Cambodia. So work by Burnet Institute group has shown that Cambodian parasites are essentially hypermutators. They have special mutations in their DNA that even allow them to mutate even more. And what they also find is when they did kind of whole genome sequence of these resistant parasites, they found they grouped into 3 distinct groups, which would suggest that there could be potentially 3 different routes towards resistance. But despite all this that we can hypothesize and explore, we actually know very little about how resistance actually works. Now that actually changed a couple of months ago when this paper was published in Nature. And they identified several mutations that they said were associated with resistance. So what they did is they did whole genome sequencing of both resistant and susceptible parasites and revealed mutations in the K13 propeller domain. Now the K stands for Kelch proteins. These Kelch proteins are found in lots of different organisms. No one really knows what they do, but they're thought to be involved in protein-to-protein interactions. But here's a schematic of it up in the top left there showing the mutations there. So what this group did is they found that these mutations were associated with both in vitro survival rates and also in vivo parasite clearance rates. So we look at the graph on the top right hand side, so the wild type parasites there in green, they're getting cleared quite rapidly from these patients, so around a mean of about 2.5 hours. Whereas parasites with the different mutations took much longer to clear from patients who were treated with artemisinin. They also found that this polymorphism also clustered in areas where resistance was confirmed. So if we have a look at the green as a wild type, you can see that up in the bio. But this red mutation, C5801, is found very high prevalence in Western Cambodia, where this resistance has emerged. Now this marker is yet to be validated, but needless to say, once it is kind of validated, it will be a very useful molecular marker for the surveillance of artemisinin resistance, both in the Greater Nepal and beyond. So what's being done in the scientific community to figure out how far this resistance is spread? Now this is where the Tracking Resistance Artemisinin collaboration comes in. And this is a study that's being coordinated by the Mahidol Oxford Research Unit and supported by DFID and Ward. So this study here is essentially 2,000 patients covering 16 study sites over 9 countries and 2 continents. So we've got study sites both in around the epicenter, further west, and also in Africa too. And there's really 2 kind of primary objectives of this study. One is to determine the spread of resistance from Western Cambodia and beyond. And the second is to identify factors that influence artemisinin sensitivity. So one would be the parasite genetic factors, like this mentioned previously. And the other is also looking at host factors. And that's what my research at the Burnet Institute does. So that's what the scientists are currently doing, but what about the global plan? So WHO launched GPARC in 2011, which is the global plan for artemisinin resistance containment. And this is a high-level plan of attack to really protect and preserve the use of artemisinin combination therapy. There are two goals. One is to contain and eliminate artemisinin resistance where it already exists. The second is to prevent artemisinin resistance where it's not yet appeared. Now, there has basically 5 prongs to this attack. One is to stop the spread of resistant parasites. 2 is to increase monitoring and surveillance, and that genetic monitoring can now be incredibly useful. 3 is to improve access to diagnostic rational treatment, and that's going back to that test treat track that Ray was talking about in his presentation before. 4 is to increase investment into artemisinin resistance-related research. And 5 is to motivate action and mobilize resources. And I think Emily really nicely described Australia's response to tackling this problem in the talk before. So essentially there's 2 important documents if you're interested in reading more about this. So one is the GPARC itself, and then also last year WHO launched its more targeted response to the equation last year. So when I kind of get back to the title of my talk, History Repeating Itself, well, one way I kind of think it is because we're once again seeing the emergence of drug-resistant parasites in the same area. But really, we've got a really good opportunity now to make sure the history doesn't repeat itself and then make sure that drug resistance doesn't spread globally. So really, we've got to kind of all get together, both as a research community and as a global community, to stop the drug resistance. So that's been my talk. I just quickly like to thank the funders and partners of my research into artemisinin resistance.